Hello students, today we will learn about polymerase chain reaction. It's a DNA amplification. We will study details techniques of polymerase chain reaction and how we can use its all the applications we will discuss in this video. So let's begin. So chain reaction. First of all, we'll know what is the main polymerase chain reaction, or what is the definition. In short, PCR technique. In biotech world, we can say as PCR technique. Okay, it's a laboratory technique we will study. Now, the polymerase chain reaction is a laboratory technique for generating large quantities of a specified DNA. Obviously, PCR is a cell-free amplification technique for synthesizing multiple identical copies of any DNA of interest. It is developed in 1984 by Curry Mullis, the scientist named Curry Mullis. He is a Nobel laureate in 1993. PCR is now considered as a basic tool for the molecular biologist. As a photocopier, a basic requirement of an office, so is the PCR machine in a molecular biology laboratory. Now let's know about the principles of PCR. What are the principles? The double stranded DNA of interest is denatured to separate into two individual strands. Each strand is then allowed to hybridize with a primer, that is renaturation. The primer template duplex is used for DNA synthesis. These three steps, denaturation, renaturation and synthesis, are repeated again and again to generate multiple forms of target DNA. Now, what is the technique? Step by step we will discuss. So, first step is the essential requirement for PCR are, first is the target DNA. The main topic is target DNA, that is 100 to 35,000 base pair in length. Second thing requirement is two primers, synthetic oligonucleotides of 17 to 30 nucleotides length that are complementary to regions lacking the target DNA. Third requirement is 4 deoxyribonucleotides, that is a DATP, DCTP, DGTP and DTTP. Fourth requirement is a DNA polymerase that can withstand at a temperature up to 95 degrees centigrade, that is thermostable. The reaction mixture contains the target DNA, two primers, a thermostable DNA polymerase that is isolated from the bacterium Thermus aquaticus, that is stacked DNA polymerase, and 4 deoxynucleotides. The actual technique of PCR involves repeated cycle for amplification of target DNA. Each cycle has 3 stages. First is denaturation, second is renaturation, and third is synthesis. Denaturation. On raising the temperature to about 95 degrees centigrade for about 1 minute, the DNA gets denatured and the two strands separate. Second is renaturation, or we can say annealing. As the temperature, the mixture is slowly cooled to about 55 degrees centigrade. The primer's base pair with the complementary regions, flaking target DNA strands. This process is called renaturation or annealing. High concentration of primer ensures annealing between each DNA strand and the primer rather than the two strands of DNA. Now let's know about the synthesis. The initiation of DNA synthesis occurs at 3' hydroxyl end of each primer. The primers are extended by joining the bases complementary to DNA strands. The synthetic process is PCR is quite comparable to the DNA replication of the leading strand. However, the temperature has to keep kept optimal as required by the enzyme DNA polymerase. For tag DNA polymerase, the optimum temperature is around 75 degrees centigrade. For E. coli DNA polymerase, it is around 37 degrees centigrade. The reaction can be stopped by raising the temperature up to about 95 degrees centigrade. The stages of PCR in relation to temperature and time you can discuss. Each cycle of PCR takes about 3 to 5 minutes. In the normal practice, the PCR is carried out in an automated machine. The new DNA strand joined to each primer is beyond the sequence that is complementary to the second primer. These new strands are referred to as long templates and they will be used in the second cycle. For the second cycle of PCR, the DNA strands, original plus newly synthesized long template, are denatured, annealed with primers and subjected to DNA synthesis. At the end of second round, long templates and short templates, DNA strands with primer sequence at one end and sequence complementary to the other end are found. The third cycle of PCR, the original DNA strands along with long and short templates are the starting materials. The technique of denaturation, renaturation and synthesis are repeated. This procedure is repeated again and again for each cycle. It is estimated that at the end of 32 cycle, 32 end cycle of PCR, about a million fold target DNA is synthesized. The short templates processing precisely the target DNA as double stranded molecules are accumulated. Now let's know about the sources of DNA polymerase. From where we get DNA polymerase. In the original technique of PCR, clino fragment, clino fragment of E. coli DNA polymerase was used. This enzyme gets denatured at higher temperature, enzyme therefore fresh enzyme had to be added for each cycle. A breakthrough occurs with the introduction of tag DNA polymerase from thermophilic bacterium, that is Thermus aquaticus. The tag DNA polymerase is heat resistant. Hence, it is not necessary to freshly add this enzyme for each cycle of PCR. Now, let's know the key factors for optimal PCR. Primer. First is primers. Primers play a significant role in determining PCR. The primers 17 to 30 nucleotides without secondary structure and without complementary among themselves are ideal. The complementary among themselves are ideal. The complementary primers are hybridized to form primer dimer and get amplified in PCR. This prevents the multiplication of target DNA. Now let's know about the DNA polymerase. Tag DNA polymerase is preferred as it can withstand high temperature. In the hot start protocol, DNA polymerase is added after the denaturation step of first cycle. This avoids the extension of the mismatched primers that usually occurs at low temperature. Tag DNA polymerase lacks proofreading exonuclease, that is 3' to 5' activity, which might contribute to errors in the products of PCR. Some other thermostable DNA polymerase with proofreading activity have been identified. Example, TMA DNA polymerase from Thermotoga marittima. PFU DNA polymerase from Pyrococcus furiosus. Now, target DNA. Let's know about the target DNA. In general, the shorter the sequence of target DNA, the better is the efficiency of PCR. However, in recent years, amplification of DNA fragments up to 10 kilobase pair has been reported. The sequence of target DNA is also important in PCR. Thus, GC-rich regions of DNA strand hinder PCR. Let's know about promoters and inhibitors. Addition of proteins such as bovine serum albumin (BSA) enhances PCR by protecting the enzyme DNA polymerase humic acid. Humic acids frequently found in archaeological samples of target DNA inhibits PCR. Now let's know about the variations of PCR. The basic techniques the basic technique of PCR has been described here in this part. Being a versatile technique, PCR is modified as per the specific demands of situation. Thus, there are many variations in original PCR. Some of we will now discuss. Some of them we will discuss. First is nested PCR. Nested PCR sequence. What is this? Sequence similarities between the target DNA and related DNA are very frequently seen. As a result of this, the primers may bind to both the DNAs and therefore even the undesired DNA also get amplified in PCR. Use of nested primers increases the specificity of PCR and selectively amplifies target DNA. Nested PCR uh, in uh, in the first cycle of PCR, the products are both from target DNA and undesired DNA. A second set of internal primers is now used. They will selectively bind to target DNA and amplification process proceeds. Now, inverse PCR. In the inverse PCR, amplification of DNA of the unknown sequence is carried out from the known sequence. The target DNA is cleaved with a restriction endonuclease which does not cut the known sequence but cuts the unknown sequence on either side. The DNA fragments so formed are inverted and get circularized. DNA ligase is employed as a sealing agent. The circle containing the known sequence is now cut with another restriction enzyme. This cleaves only the known sequence. The target DNA so formed contains the known sequence at both the ends with target DNA at the middle. The PCR amplification can now be carried out. It may be noted that the primers are generated in the opposite direction to the normal since the original sequence is inverted during circularization. Now, let's know about anchored PCR. In the anchored PCR, a small sequence of nucleotides can be attached to the target DNA. That is, the DNA is anchored. This is particularly useful when the sequence surrounding the target DNA is not known. The anchor is frequently a poly G tail to which a poly C primer is used. The anchoring can also be done by the use of adapters. As the adapters possess a known sequence, the primer can be chosen. Now let's know about the reverse transcription PCR. RT-PCR, in short, RT-PCR. What do we mean by that? Let's know about this. The PCR technique can also be employed for the amplification of RNA molecules, in which case it is referred to as a reverse transcription. PCR, that is RT-PCR. For this purpose, the RNA molecule must be first converted to complementary DNA by the enzyme reverse transcriptase. The cDNA then serves as the template for PCR. Different primers can be employed for the synthesis of DNA. For the synthesis of first strand of cDNA. These include the use of random primers, oligo DT uh, primer, and a sequence specific primer. Now let's know about the asymmetric PCR. PCR technique can also be used for the synthesis of single stranded DNA molecules, particularly useful for DNA sequencing. In the asymmetric PCR, two primers in a ratio of 100 is to 1 are used. After 20 to 25 cycles of PCR, one primer is exhausted. The result is that in the next 5 to 10 PCR cycles, only single stranded DNAs are generated. Now let's know about the real-time quantitative PCR. The quantification of PCR products in different cycles is not as simple as projected by theoretical considerations. In practice, large variations occurs. The most commonly used technique for measuring the quantity of PCR is by employing a fluorescence compound like ethidium bromide. The principle is that the double-stranded DNA molecules bind to ethidium bromide, which emit fluorescence that can be detected and DNA quantified. The synthesis of genes by PCR and the role of PCR inside directed mutagenesis is now we will discuss. Now let's know about the random amplified polymorphic DNA. In short, RAPD, random amplified polymorphic DNA. Normally, the objective of PCR is to generate defined fragments of DNA from highly specific primers. In the case of RAPD, pronounced as rapid, in short, oligonucleotides primers are arbitrarily selected to amplify a set of DNA fragments randomly distributed throughout the genome. This technique, random amplified polymorphic DNA, is also known as arbitrarily primed PCR. In short, AP-PCR. The procedure of RAPD is comparable to the general technique of PCR. This method basically involves the use of a single primer at low stringency. A single short oligonucleotide binds to many sites in the genome and the DNA fragments are amplified from them. The stringency of primer binding can be increased after a few PCR cycles. This allows the amplification of best matches. RAPD can be carefully designed so that it finally yields genome specific band patterns that are useful for comparative analysis. This is possible since genomic DNA from two different individuals often produces different amplified patterns by RAPD. Thus, a particular DNA fragment may be generated for one individual and not for the other, and this represents DNA polymorphism, which can be used as a genetic marker. Now, let's know about the RAPD let's know about the RAPD in brief. RAPD is widely used by plant molecular biologists for the genetic identification of plant species. For this purpose, different combinations of nucleotides, most of them random oligonucleotide primers, have been designed and are commercially available. As each random primer anneals to a different region of DNA, many different regions of loci on the DNA can be identified. RAPD is thus useful for the construction of genetic maps as a method for genomic fingerprinting. Now let's know about the limitations of RAPD. The main problem of RAPD is associated with reproducibility. It is often difficult to obtain similar levels of primer binding in different experiments. It is therefore difficult to correlate results obtained by different research groups on RAPD. Now, let's know about AFLP. What do you mean by AFLP? Amplified fragment length polymorphism. Amplified fragment length polymorphism, in short AFLP. Let's know about this. FLP is a very sensitive method for detecting polymorphism in the genome. It is based on the principle of restriction fragment length polymorphism and RAPD. FLP may be appropriately regarded as a diagnostic fingerprinting technique that detects genomic restriction fragments. In the AFLP, PCR amplification rather than southern blotting is used for the detection of restriction fragments. It may be noted that AFLP is employed to detect the presence or absence of restriction fragments and not the lengths of these fragments. This is the major difference between FLP and RFLP. FLP is very widely used in plant genetics. It has not proved useful in the mapping of animal genomes since this technique is mainly based on the presence of high rates of substitutional variations which are not found in animals. On the other hand, substitutional variations resulting in RFLPs are more common in plants. The basic principles of AFLP involves the amplification subsets of RFLPs using PCR. A genomic DNA is isolated and digested simultaneously with two different restriction endonucleases. EcoR1 with a 6 base pair recognition site and M-cell with a 4 base pair recognition site. These two enzymes can cleave the DNA and results in small fragment that is that is lower than 1 kilobase pair, which can be amplified by PCR. For this purpose, the DNA fragments are ligated with EcoR1 and M-cell adapters. These common adapter sequences that is plaquing genomic sequences serve as primers, each having only one selective nucleotides. These PCR products are diluted and used as template for the selective amplification, employing two new AFLP primers that have two or three selective nucleotides. After the selective amplification by PCR, the DNA products are separated on a gel. The resultant DNA fingerprint is identified by autoradiography. AFLP fragments represent unique positions in the genomes and hence can be used as landmarks to bridge the gaps between genetic and physical maps of genomes. In plants, AFLP is useful to generate high density maps and to detect genomic clones. Now, let's know about RACE. RACE, in brief, rapid amplification of cDNA ends. Rapid amplification of cDNA ends. In short, RACE. Reverse transcription followed by PCR results in the amplification of RNA sequences in cDNA form. But the major limitation of RT-PCR is related to incomplete DNA sequences in cDNA. This problem is solved by using the technique rapid amplification of cDNA ends. The target RNA is converted into a partial cDNA by extension of a DNA primer. This DNA primer was first annealed at an interval position of RNA not too far from the 5' end of the new molecule. Now addition D and ATP and terminal deoxynucleotidyl transferase extends the 3' end of the cDNA. This happens due to the addition of a series of R's to the cDNA. This R series now acts as the primer to anneal to the anchor primer. A second strand of DNA can be formed by extending the anchor primer. The double-stranded DNA is now ready for the amplification by PCR. This procedure is called 5' race since it is carried out by the amplification of the 5' end of the starting RNA. Similar protocol can be used to carry out 3' race when the 3' end RNA sequence is desired. Now let's know about the limitations of race, RACE. Since a specific primer is used, specificity of amplification of rays may not be very high. Another disadvantage is that the reverse transcriptase may not fully reach the 5' end of RNA, and this limits the utility of rays. In recent years, some modifications have been done to improve rays. Now let's know about the applications of PCR. The advent of PCR had and continues to have tremendous impact on molecular biology. The applications of PCR are too many to be listed here. Some of them are selectively and very briefly described here. Now, PCR in clinical diagnosis. How PCR is used in clinical diagnosis. The specificity and sensitivity of PCR is highly useful for the diagnosis of various diseases in humans. These include diagnosis of inherited disorders, viral diseases, bacterial diseases, etc. The occurrence of genetic diseases frequently identified by restriction fragment length polymorphism, that is RFLP, can be employed only when there is a mutation resulting in a detectable change in the length of restriction fragment. Many genetic diseases occur without the involvement of RFLP. For all disorders, PCR technique is a real boon as it provides direct information of DNA. This is done by amplification of DNA of the relevant region, followed by the direct analysis of PCR technique, PCR products. Now, let's know about the prenatal diagnosis of inherited diseases. How uh, we can search by how you can use diagnose prenatal diseases, inherited diseases by the help of PCR. PCR is employed in the prenatal diagnosis of inherited diseases by using chorionic villus samples or cells from amniocentesis. Thus, diseases like sickle cell anemia, beta thalassemia, and phenylketonuria can be detected by PCR in these samples. Now, let's know about diagnosis of retroviral infections. PCR from cDNA is a valuable tool for diagnosis and monitoring of retroviral infections, example HIV infection. Now let's know about the diagnosis of bacterial infections. PCR is used for the detection of bacterial infections, example tuberculosis by Mycobacterium tuberculosis. Now let's know about the diagnosis of cancers. Several virally induced cancers, example cervical cancer caused by human papilloma virus, can be detected by PCR. Further, some cancers which occur due to chromosomal translocations, chromosome 14 and 18 in follicular lymphoma involving known genes are identified by PCR. PCR in sex determination of embryos. Sex of human and livestock embryos fertilized in vitro can be determined by PCR by using primers and DNA probes specific for sex chromosomes. Further, this technique is also useful to detect sex-linked disorders in fertilized embryos. Now let's know about the PCR in DNA sequencing. As the PCR technique is much simpler and quicker to amplify the DNA, it is conveniently used for sequencing. For this purpose, single strands of DNA are required. In asymmetric PCR, preferential amplification of a single strand is carried out. In another method, strand removal can be achieved by digesting one strand. Now let's know about the PCR in gene manipulation and expression studies. The advantage with PCR is that the primers need not have complementary sequences for the target DNA. Therefore, the sequence of nucleotides in a piece of a gene, that is target DNA, can be manipulated and amplified by PCR. By using this method, coding sequence can be altered to synthesize protein of interest. Further, gene manipulations are important in understanding the effects of promoters, initiators, etc. in gene expression. PCR is important in the study of mRNAs, the products of gene expression. This is carried out by reverse transcription, PCR. PCR in comparative studies of genomes. The difference in the genome of two organisms can be measured by PCR with random primers. The products are separated by electrophoresis for comparative identification. Two genomes from closely related organisms are expected to yield more similar bands. PCR is very important in the study evolutionary biology, more specifically referred to as phylogenetics, as a technique which can amplify even minute quantities of DNA from any sources, hair, mummified tissues, bone, or any fossilized material. PCR has revolutionized the studies in paleontology and archaeology. The movie Jurassic Park has created public awareness of the potential application of PCR. PCR in forensic medicine. PCR in forensic medicine. A single molecule of DNA from any sources, that is blood stain, hair, semen, etc. of an individual is adequate for amplification by PCR. Thus, PCR is very important for identification of criminals. PCR in comparison with gene cloning. How PCR is used for gene cloning. The PCR has several advantages over the traditional gene cloning technique. These include better efficiency, minute quantities of starting material, that is DNA, cost effectiveness, minimal technical skill, time factors, etc. In due courses of time, PCR may take over most of the applications of gene cloning. In this video, I have completed PCR technique, all the applications also I have cleared. Any questions regarding this PCR, you can ask me in the comment box, and subscribe my channel to know more topics about biotechnology. Thank you.